Hello everyone, my name is Aklesh Teli, Master Integrated PhD student from SL Lab under the supervision of Jiyang Park. Today I am going to talk about the specific application of radio frequency wave biosensor technology, and my today outlines are determination of a glucose in the blood using amperometric biosensor. Then we will discuss about the determination of a nanogram level of a copper in water using anodic stripping voltammetry employing an electrode modified with complexing agent. Then we will discuss about the determination of the cellular ion. Simultaneously, we will also talk the laboratory chips. Then we will go through the determination of the atmol level of a tiny troline complex with luminescent transducer, and last we will discuss about the determination of the flavonols in a beer. After discussing all these topics, we will conclude our presentation with the conclusion. Now, determination of the glucose in a blood using amperometric biosensor. There are number of different ways of determining glucose just by using electrochemical transducer. Glucose oxide lies at the center of all of them, but here we will talk about the amperometric biosensor. Now for this, let's provide a simple portable sensor for you so that we can use at the home by the diabetic patient for the regular monitoring of our glucose level in their own blood, and also it is very useful by the patient so that they can use at the home for the regular interval, and it is stable over a long period of time, especially when they are sealed in a foil. Now, the determination of a nanogram level of a copper in water using anodic stripping voltammetry, then employing an electrode with modified complexing agent. Anodic and cathodic, these are the two different electrodes. This process can be improved if the material is absorbed onto the surface in some way. This is the basis of a technique for analyzing low concentration of a metal ion such as a copper. On the left hand side, we can see the typical anodic stripping and cathodic we are using. Now, for to make an ultra sensitive sensor so that it will detect very low level of a copper's ion, we need to require some agent such that 2,9 dimethyl 1,1 will to form the complex with the copper. A voltammetric electrode is used in this case. Now the carbon paste entrapped method is very suitable for this particular application, and the concentration time is also very low, that is 1 second. It's very high, that is 1 second to 3 minutes. required. So the electrode holder was made from a plastic syringe tube with an electrode area of 0.02 centimeter square. Now this method was tested against the NBS, National Bureau of Standards. Now the determination of the several ions simultaneously in the lab, then chemiregisters are introduced. The sensor based purely on variation of the resistance of a sensor in the presence of an analyte generally lacks sensitivity. Each sensing element can be coated with a different conducting polymer or made from the synthode metal oxide. Now the data obtained can be used to construct non-parametric nonlinear model of any array response. Now the sensor array and the smart sensor. For that, integrated signal processing eliminate electronic noise, stray capacitance and electromagnetic interference, thus improving the signal to the noise ratio. They can also be automatic calibration and the temperature correction. On left hand side we can see the schematic representation. Determination of atmol level of trinitrotoluene antibody complex with an luminescent transducer. Background to immunoluminescent assays. Certain biological species, principally the firefly, can be emit luminescence. The enzymes catalyze oxidation of a luciferin result in luminance as follow. We can see in the figure left hand side there is the figure, the reaction schematic of the luminance assay of a. Now for that we need to detect very low level of explosive resins in the environmental situation. Now the determination of a flavonols in beer. For that, banana is developed for the determination of a doping, that is a brain chemical. The polyphenol oxides catalyze the oxidation of the cathols by the ambient from the ambient oxygen into the ocunes. This can then be used to determine certain cathols type substance, and also this is known as the phenols, which are found in beer and wines. On the left hand side we can see the schematic catalyst of the cathols by the polyphenol oxides. Now at last we will conclude our conclusion. In this presentation slide we will first discuss about the number of the sensor technique, then we will also see the structure and the performance of the Medimans extract, and then we will also go through the low concentration of a copper in a water and analyze by complexing and the different pulse polarography, and last we will talk about the fourth example that describe how the femtomole amount of explosive material can be determined by the combination of the amino acids linked by the enzyme process to a bioluminescent assay. In this we will see the example of the firebird. Thank you.